Buon pomeriggio a tutti, grazie per la vostra partecipazione, grazie in particolare al professor VLADIMIR MUZYKANTOV per essere con noi. Alcune parole in italiano, poi alcune in inglese, a beneficio di tutti. Il professor MUZYKANTOV ha accettato di tenere questo seminario qui all'Università di Urbino, che è organizzato nell'ambito sia del dottorato in Biochemical and Health Science, facoltà anche di Scienze Biologiche e Biotecnologie, e anche in collaborazione con un'altra iniziativa che è quella del progetto Marche Biobank. Lo scopo di questo seminario è per introdurre una tematica che è comune a molti ricercatori dell'Università di Urbino e che è particolarmente attiva come approccio e come interessi, soprattutto negli ultimi anni, che è quello sostanzialmente del target delivery, cioè stiamo passando da una medicina dove il farmaco viene somministrato e va bene per qualsiasi comparto ad una medicina dove il farmaco è personalizzato insieme alla terapia e anche diretto verso il distretto, l'organo, la cellula di interesse dove deve svolgere la sua funzione. Few words about Vladimir. We are colleagues and friends since many years. Vladimir is professor of pharmacology, but as a matter of fact, in the US, the situation is a little bit different from Italy. In Italy, we belong to a single department. This is not the case from the US. In fact, Vlad is a member of the Institute for Medical Engineering, is a member of the Center for Cancer Pharmacology, is a member for the Cardiovascular Institute, and several others. Of special interest, and I would like to underline this point, Vlad was the founder and co-director of the Center for Targeted Therapeutics and Translational Medicine. This gives you an idea of what the interests of Vlad's are. A center dedicated to this kind of activities. Vlad has, sorry Vlad, I will summarize, and certainly I will forget a huge number of activities we are running actually, and they're recent fast. For our benefit, the research is essentially focused on two main areas. One is the definition of specific targets. If you want to do a targeting approach, you need to know what's the molecule you want to target for a specific delivery or for a specific function. And he is working on the discovery of system that permits to take advantage for some molecules that are present, expressed in physiological or pathological condition, especially on the vascular system. Not in general, because as Vlad will tell us, the vascular system is not exactly expressing the same biomarker or molecules if we are talking about the lung or the heart or the central nervous system. Second area of interest is the area that permitted years ago to connect each other, that is the possibility of exploring red blood cells as a delivery system. This is with respect to what we are doing in Urbino, based on, I will say, a different approach that is based on the possibility of having something on the surface of the cells that is able to recognize the target. This means that most of the paper and the patents Vlad has done in these years are connected to the construction of cell-based delivery and targeting system, developing also totally new molecules, totally new modalities, and totally new drugs to permit to have this specific target. So because of this and because of the implication of this kind of activities, Vlad is also inventor in a large number of patents, try to figure out, looking to database, but I'm sorry, we're too many to identify all, and is also working in collaboration with a number of companies as a scientific advisor or member of scientific committee in a number of companies that are interested in developing this kind of technology. So I think it's a great opportunity for us. I really hope you enjoy having with us Vlad today, and that can be beneficial for what you're doing actually in your area of research, and if not directly involved in the same approaches, I think you can really appreciate how important is the approach Vlad is using in doing its own activities, its own research. So Vlad, thank you very much for spending some time with us, and thank you very much for coming again to Urbino once more.
Thank you very much, Mauro. Thank you, guys. I'm speechless. I'm so grateful to Mauro for his friendship and for his wonderful words and for inviting me here today. I will try to talk in a very informal way, and that's why I'm sitting, just not to disturb my left knee. Please, if you feel like asking a question, do. Because to me, it might be much more productive and better to stuck with the slide number 2, provoking your questions and discussion, than go through all slides without any comments. So please do this, interrupt me. I will try to tell you 2 stories today, and both of them, as Mauro alluded, they are some sort of stories of my life. And that's why there are so many people and so many names on the most important slide of this presentation. These are makeup of my lab across the years. It's not all the people who are contributed to these studies are mentioned, especially in collaborators section, but these are people who I am very grateful to. Drug delivery is a very simple thing in theory. You want to deliver drug or a carrier which is filled with a drug into the organ of your interest or a cell of your interest. So you define the molecule on this cell, take an antibody or other ligand which binds to this molecule, and use this as a carrier for your drugs. Paul Ehrlich, who got a Nobel Prize with Ilya Mechnikov in 1908 for discovery of immunoglobulins, proposed this as a magic bullet. So you take an antibody against something you want to deliver to, conjugate with a drug, and it goes. You can use carriers, liposomes, polymeric particles, which would be protecting drugs from the body and body from the drugs on the route. Now, each target, each cell in our body is different in terms of targeting. It's different because they all have different functions, they have different locations, they have different determinants, and they all differ in the pathology versus quiescence state. My favorite cell in the body is endothelial cell. When I was in the medical school in early 70s, the endothelium was viewed as just an inert, sort of lubricating layer of the vessel. In reality, endothelial cells, which line lumen of the vascular system, they have tremendous functions. You can see from the list on the right what functions they control. And there is nothing in pathology or in health state which endothelial cells would not be involved in a very, very active state. They, as I say, they line the vascular wall. They form this oriented in a space pattern when they are exposed to the flow in the arteries. And generally, endothelium is a very important target involved in many diseases. Now, it's very accessible, which is good. But drugs mostly do not bind to endothelium in any specific way. So the solution seemed to be very simple. Take Paul Ehrlich's axiom and conjugate drugs with certain molecules which would bind, deliver to endothelial cells. So this is a story which begins in early 80s, and the story was opened by two papers. The first paper was from Steve Cannell, Liv Huang, and Vladimir Torchilin in 1989. They took rat lungs, homogenized, immunized a mouse, took monoclonal antibodies, stained the Western blotting analysis, and found an antibody which bound very specifically to homogenate from the lungs, not from other organs. They took this antibody, radio-labeled, injected intravenously in a mouse, in a rat, and found that it accumulates in the lungs very nicely versus immunoglobulin. Then they took liposomes, coated with this antibody, and liposomes went to the lungs very effectively. It was a wonderful discovery, but then they identified the protein. And this glycoprotein turns out to be thrombomodulin, TM. Thrombomodulin is discovered by about 10 years before this work. Thrombomodulin is indeed endothelial glycoprotein, very specific for endothelium. It's differently expressed in the various areas, but generally it's endothelial glycoprotein. The problem is that thrombomodulin has a very important function. It binds thrombin, and it converts thrombin from pro-inflammatory, pro-coagulant, pro sort of injury agent into protective agent. And you cannot fool with thrombomodulin because you predispose to thrombosis and inflammation. And this was end of the story for thrombomodulin drug targeting. I will tell about thrombomodulin more just because its functions in endothelial cells are so important that we try to intervene. But we, and by we I mean my friend Sergei Danilov and I, in Moscow in the same year published a paper on ACE. And you all know what ACE now is, but you know about ACE2. But ACE1 is a senior brother of ACE2. It's an endothelial glycoprotein which converts angiotensin 1 into angiotensin 2. Angiotensin 2 is a very potent agent for the blood pressure. It induces contraction of smooth muscles in the vascular wall. So ACE function is relatively well understood because ACE inhibitors, exactly by the 70s, become a very potent hypotensive drug. So inhibition of ACE was sort of something which could even be helpful in some situations. So instead of following the chance, we isolated, we immunized mice too with ACE, with human ACE, with rat ACE, and get an antibody. And you can see that when we injected this radiolabeled antibody in intravenous in rats, we had huge uptake in the lungs, which could be seen by gamma camera too. Now, and ACE is still in the works, but it's not, by no means it's just these two determinants which can be used for endothelial targeting. In addition to ACE, there is aminopeptidase B from KVLR, cell adhesion molecules, PCAM molecule, which is inside in the intercellular junctions. These are normal conditions. In the pathology, in inflammation, in stroke, in infarction, in tumor growth, in diabetes, all bets are off. ACE, for example, is shed. Selectins are exposed. ICAM is redistributed. PCAM is more exposed to the circulating agents. So pathological vasculature, pathological endothelial cell is very different. Now, it's not always goes in the direction of exposure. For example, as I mentioned, ACE is shed by endothelial cells in inflammation. When you inject endotoxemia in the rats, you see that the accumulation of the antibody in the lung goes down. You have to also take into account that even in the most sort of uniformly vascularized tissues, such as lungs, there is very big heterogeneity between arterial, capillary, venous endothelium. Also, between the upper lobes and lower lobes, because they are differently circulated, etc., etc. So you have to be cognizant of these differences. And what's interesting, for example, and still not explained, both thrombomodulin antibody and ACE antibody, accumulation in the lung goes down when you treat animals with endotoxin, for example. But if you take nanoparticles, 100 nanometer nanoparticles, and coat these antibodies, you see that in pathology, nanoparticle uptake is going up. We don't know the reason. Do you have any idea how it can be explained? Please. You see that the antibody itself goes down, whereas the nanoparticle uptake goes up. Well, we don't know, but the only explanation which I can come up with, which is not tested yet, is that maybe when accessibility of these determinants, thrombomodulin versus ACE, accessibility for antibodies is high, both normally and pathologically. But accessibility for 100 nanometer particles is high only in pathology when you have pathological opening and edema, etc., etc. These are main heroes of my endothelial targeting saga. These are transmembrane glycoproteins, cell adhesion molecules. PCAM is platelet endothelial cell adhesion molecule. ICAM is intracellular cell adhesion molecule. I also work with VCAM and other. These are endothelial molecules which are exposed in the lumen, and they are involved in transmigration of leukocytes and some other cells from the blood to the tissue in the sites of pathology, inflammation, for example. So theoretically, blocking their function could be protective or helpful in treatment of inflammation, thrombosis, and these are more or less common characteristics for any of these antibodies. Another thing is that the lungs represent a very privileged and specific target for vascular delivery. The lungs' surface area of endothelium is the biggest in the body. The lung receives the whole venous cardiac output, 50% of total cardiac output, and plus it receives some arterial blood output. It is perfused very slowly because this very high capacity, low resistance capillary bed for exchange of gases. So all these circulatory and surface vascular surface features make lung by default a very unique target for anything which has affinity to endothelial cells. We use this feature for example, targeting of antioxidant enzymes, SOD, superoxide dismutase, or catalase. And I'm not going to go over each graph because that will stop me at the third slide. These data are mostly published. And if you have questions concerning these technicalities, we always can go into this, but I want you to ask me any questions, technicalities, theory, anything. Please, please. Let's be proactive. So, for example, ICAM antibody, its level uptake in the organs with inflammation, thrombosis, ischemic perfusion goes up significantly. PCAM is lavishly expressed in the normal endothelial cells by 1 million copies per cell. They are very robust targets. And you can see that in that injection of isotope-labeled ICAM antibody, in contrast to FDG, which goes to the heart and goes to the fat tissue, with ICAM, you can see staining, not staining, signal of isotope from the lungs. Thrombosis, and we will come back with the example of thrombomodeling, it's a very important pathology, still not really well managed. And for example, some antithrombotic agents like plasminogen activators, TPA, urokinase, they are also very dangerous. They dissolve fibrin clot, but they cause lots of hemorrhagic changes and pro-inflammatory changes. So even a long time before us, other people were working with fusion active domains of TPA or thrombomodeling with single-chain domains of antibodies to fibrin, activated platelets, etc., etc. And they made these constructs which bound to activated platelets and induced fibrinolysis. Well, in vivo it didn't work very well because, as you can see, their targets for these fusions get very quickly masked. They are very quickly getting impervious and inaccessible to the blood elements. So what Bissen did in my lab when he was here, we took anti-CAM antibodies, made single chains, for example, PCAM single chains, and fused with anti-thrombotic agents such as plasminogen activators, or thrombomodeling itself. And they, if the endothelial cells lost normal TM, these fusions bound to pro-inflammatory molecules, to PCAM, ICAM, and produced anti-inflammatory and anti-thrombotic action over there. We had very nice controls in with animals which do not have human PCAM, for example. This is control for the mouse, normal mouse, which just lacks expression of human antigen. This is with human antigen. You see how specific and effective accumulation of the radio-labeled fusions is in the lungs. And these are controlled for non-targeted fusions. And if you inject fusions versus just soluble thrombomodeling and stain with an antibody against flag, you can see that the lung tissue is stained with the fusion, but not with soluble TM. Weak adherin is endothelial target, endothelial marker, and you can see that the staining of fusion coincides with endothelium. Ecadherin is epithelium. It's in the airways. There is no colocalization. And now if you merge these stainings, you can see that staining with fusion coincides with vascular staining, but not with epithelial staining. So on the level of tissue specificity of the cell, we have very nice delivery. Now, I'm asking again, any questions? Maybe something very general. I will be glad to. I mean, I know this stuff. So this data, which we don't go to decipher, just shows that when you inject single chain targeted to PCAM and fused with thrombomodulin, you completely replenish lost function of thrombomodulin in animals with ischemic perfusion, endotoxemia, ischemia, etc. Now, thrombomodulin is a very interesting creature. It doesn't have any activities unless it binds thrombin. But when it binds thrombin, it interacts with another protein, EPCR, endothelial protein C receptor, which collaborates with thrombin in providing APC, activated protein C. It's very potent anti-inflammatory, anti-edematose, anti-thrombotic agent, endogenous. The problem is that PCAM, as you can see, is localized predominantly in the intercellular junctions in the endothelial cells. And EPCR there's no super staining. ICAM is expressed predominantly in the apical body, apical membrane of endothelial cells, and it coincides with EPCR. So when we, instead of targeting to PCAM, targeted to ICAM, we capitalized on this natural proximity, and effects were even more effective. Now, this is a very enigmatic and still unexplained phenomenon, which I just briefly mentioned. Then, if you have PCAM antibody, which binds to PCAM in vivo or in vitro, and add another antibody, which binds to a different epitope on the PCAM, you see an increase twofold, threefold in vivo, 100-fold times increase in binding to the PCAM of this labeled antibody. We deciphered this in a way that the first antibody induces the conformational changes in the PCAM molecule, which open up additional binding sites for the second antibody. This is, I work with Drew a lot. You all, I'm sure you all know who Drew Weissman is. And about 7 years ago, he asked me, why wouldn't we do LNP RNA delivery system? And we conjugated with LNP, anti-PCAM, anti-ICAM, IgG, or this is control, or targeted. And you can see that, lo and behold, a localization of LNP radio-labeled in the lungs goes orders of magnitude up. And this leads to wonderful expression of luciferase tracer in these animals. And the ratio between this and this, its liver, goes up 24 times. So that was just a tracer, luciferase. So now we have VCAM antibody. Inject radio-labeled antibody to VCAM, and we see that in the lungs, there's almost no uptake, which is strange, but it's fact. In the brain, there's a lot of uptake. The same for liposomes, and the same for LNPs. VCAM targeted anything goes to the brain. And if you have an inflammation in the brain, if you inject TNF, for example, in the hemisphere, the increased VCAM uptake, like 10 times more. Nothing else, no transferrin receptor, nothing else, even close comes to this result. And here you can see that it's not a mechanical embolization. It's staining of the vasculature in the mice, especially with inflammation. And that can be used for prevention of blood-brain barrier leakage. And blood-brain barrier is well-known, difficult target in the body. But it's difficult target because people try to overcome it. They try to deliver things beyond the BBB, which is a very difficult proposition. We do a different thing. We deliver the mRNA, which encodes thrombomodulin, to the BBB, to the endothelial cells in the injured brain. And you can see that we deliver enough to reduce edema in the brain very effectively. And this is, again, this is thrombomodulin. But this is thrombomodulin, not a protein. This is thrombomodulin mRNA. And now you must ask me a question. Vladimir, how can you deliver thrombomodulin in one case to the surface of endothelial cells, and in another case, mRNA into the endothelial cells, using the same antigen, PCAM? So you ask me, and it will be difficult to answer, except for the fact that we found that PCAM is a very interesting antigen. When you have a monomolecular ligand, antibody, or single chain fusion, which binds at a single binding site of the PCAM and on endothelial cells. Endothelial cells do not internalize it. That's why when we inject thrombomodulin fusion, which binds to PCAM, we see direct and immediate effect of thrombomodulin anchored to the surface. But if we have nanocarriers, liposomes, LNPs, polymeric nanoparticles, or just conjugated multiple antibodies against the same epitope, that induces crazy active endocytosis of endothelial cells. Sylvia characterized and decoded the mechanism of this endocytosis. It's not a canonical endocytosis by any rate. It's not caveoli, not clathrin, nothing. It's not phagocytosis. It's a very unusual uptake, which is working using non-internalizable, like anchoring molecule. And it happens not just in vitro, but you can see that in the lungs, these 100 micrometer particles go in the endothelial cells. This is with Samir. We tested effects of the shape on this in vivo. To make story even more intriguing, if you take different antibodies against domains of PCAM extracellular part, they all will bind. The antibody to 6 will bind the worst because it's too close to the plasma lemma. But the particles will bind differently. And in fact, the particles which targets to this domain will not bind at all because it's too close to the plasma lemma. It's too masked by the molecule itself. Now, if you now see green are internalized particles, yellow are non-internalized particles. You see that amongst these 5 antibodies which bind, 3 get internalized nanoparticles and 1 does not. Why? I don't know. But even further, the itinerary of intracellular trafficking of these particles, which enter in via the same non-internalizable normal anchor, they traffic into lysosomes is totally different. Further, the geometry of these conjugates, size, first of all, modulate and control the uptake. If you have too large particles or conjugates like micron, they don't get into the cells. If you have a shape irregular, like protein polyplexes, they will get into the cell if you have them small, but not large. And if you have the particle, spheroid, evenly coated with antibodies, they get into the cells, even 5 micron, even erythrocyte. If you conjugate erythrocyte to antibodies to PCAM or ICAM, they will get into endothelial cells. And you can use this, you can see that one antigen, PCAM, provides you such a large roster of opportunities, of options, where to go, how to go, which material will stay on the surface, which material will get into the cell, which material will get from endosome relatively quickly. So these are all targets, not all, many targets else, which we use. These are cargoes, including catalase, SOD, RNA, and these are delivery conjugates we use as a sort of an arsenal for different delivery paradigms in different situations. For example, reactive oxygen species produced into the lumen of endosomes are very important cellular messenger systems, inducing activation of NF-kappa B, etc. When we deliver SOD to specific types of endosomes, we very effectively inhibit this signaling. Caveoli are very special. My friend Jan Schnitzer published this paper in Nature 20 years ago, in which he showed that caveoli antigens used for targeting, for anchoring of specific ligands, deliver these ligands across the endothelial cells to the tissues. And we worked with ferritin nanocarriers targeted to caveoli, and we see very nice uptake in the lungs. And then we found that depending on the way you structurize your antibody and your cargo, SOD, in these particles, you either have a very nice in vivo delivery and effect, or you have none. Because if you have both SOD and antibody on the surface, affinity is not sufficient. So that's just data. And again, this is more of a cartoons, but, well, first of all, it shows this beautiful fenestrae, diaphragm of fenestra. This cartoon shows that depending on the route of the intracellular delivery, you may find different effects of the same cargo. This is a very interesting in vivo experiment. We injected PV1, which is caveolar antigen antibody, conjugated with polystyrene beads versus fluffy polymeric nanogel. And here we see almost no lung uptake because there is no accessibility of PV1 here to the rigid nanoparticle circulating in the bloodstream. But fluffy polymeric nanogels, even 300 nanometers, accumulate in the lungs because they can squeeze into the small openings of the caveoli. So these are just to reiterate the variability, the flexibility of the system for delivery of things into the endothelial cells. But of course, it's not all that simple. For example, many of these delivery systems might induce unintended effects in endothelial cells, including their injury or pathological activation. For example, if you have nanocarrier which aggregates very quickly in the plasma, in the bloodstream, they will be mechanically interrupted by microvasculature, which is pro-inflammatory. And here we have the last 5 minutes to address the first lap of my scientific life, which I started work 40 years ago. And red blood cell, essentially, it's a Mother Nature produced carriers. They have no other function in the body. They are just cute red things, very, very elastic, which circulate for 3 months and deliver gases and some other things from one place to another. So my friend Mauro used red blood cells for loading diverse agents, drugs, which he then re-injects, reinfuses, and their pharmacokinetics and the activity in the body is completely different from the soluble agents. In order to be not entirely like him, I was working from the very beginning on coupling drugs to the surface of red blood cells. It might look sort of counterintuitive, but in reality, red blood cells transfer lots of things on this surface. For example, CR1, it's a complement receptor 1 on red blood cells. It binds immune complexes in blood and delivers them to the macrophages in the spleen and the liver, where these macrophages take this harmful stuff from the red blood cells without damaging red blood cell. It's a very interesting thing. So there are many ways to bind or to conjugate things to red blood cells, to the surface. Unfortunately, Rubius, who I included in this slide a couple of years ago, went belly up by technical reasons. But what we show here, that when we label red blood cell carrier by chromium and cargo, which are thrombomodulin, plasminogen activators by iodine, you see that red blood cells circulate normally, but they prolong circulation of the cargo by orders of magnitude. So you can inject a fusion protein, essentially the same thing as to endothelium, just to red blood cell. You load red blood cells with this prodrug, which is activated by thrombin, and when it circulates and it gets included in the active thrombosis, it dissolves the thromb. And this data show that it works in vivo, in mice and in rats with a model of thrombotic stroke. RBC can be loaded, of course, by many approaches. You can even inject them differently, IV, with different pace, and they will distribute differently. For example, if you inject very quickly, only a very small fraction of RBC will be coated by the drug, and that will be a heavy coat. If you infuse it slowly, the same drug, the same dose, almost every red blood cell in the body will be painted by the drug at a very relatively low per RBC dose. And you can indeed use many different maneuvers to load red blood cells. What is important, that surface loading can be done without taking red blood cells out of the body. You can just make a fusion protein which binds to red blood cells, and we do this by phage display. We found that there are some ligands for red blood cells which do not damage red blood cell, don't make them more fragile or rigid. They circulate nicely. They may be used for loading nanocarriers to red blood cells just by injecting of RBC-targeted liposome or nanoparticle. It will bind to red blood cells in vivo like that, and you can test their biocompatibility, and in some cases biocompatibility suffer a lot. For example, if you have polystyrene beads targeted to red blood cells, they damage red blood cells. But liposomes and nanogels don't, and you can use this to combine two major approaches: non-targeted physical or electrostatical interactions with your targeting ligands. How do you do this? You use nanoparticles, which you load on red blood cells, or you inject in the bloodstream and they load red blood cells in vivo, and then you inject this in the vasculature of interest. Chromium labeled RBC. Nanocarrier bound RBC circulate the same way. There's no changes, no damage to red blood cells. Now here we have the same formulation traced by iodine labeling the liposome or the nanocarrier. You can see everything is in the lung. This is transfer. This is momentarily transfer of radio-labeled polymer carrier bound to red blood cell from blood to lungs. If you inject intraarterially in the carotid artery, it happens in the brain. Now, if you take, these are just nanoparticles. But if you take nanoparticles which are coated with antibody to endothelial cells, in this case PCAM, and you compare, then believe me, chromium label always the same, high in blood and nothing in other organs. Concentrate on lungs. These are just blind nanocarriers. These are nanocarriers, the same, loaded on red blood cells. These are nanocarriers with antibodies to PCAM. These are nanocarriers with antibodies to PCAM, loaded on the red blood cells. By combining these two principles, you have 1,000 times higher uptake in the pulmonary microvasculature. And two last slides. I'm not going even to try to comment this specifically, but if you take a liposome and cover it with a mixture of antibodies, one antibody against red blood cell for anchoring to the carrier, and another antibody to the endothelial antigen, which appears in the pathology or even normally like PCAM. And if you modulate the ratio between those two antibodies properly and avoid hyperavidity to red blood cells, because red blood cells are huge capacity. If you have more than 5 antibodies per liposome and inject in the body, they will suck all liposomes. So you have just 4 antibodies to red blood cells and 90 or 100 antibodies against PCAM or ICAM and inject. Then you can see that this dual targeted red blood cell accumulate in the lungs very nicely. These are single targeted. And chromium labeled red blood cells are retained in the bloodstream. This is for PCAM. This is the same for ICAM with the difference on the expression. So this is the very last slide. And this is for my friend Maura and for Sarah. The cell and biological and hybrid therapies, carriers, are very exciting story. And of course, the use of exosomes and other vesicles in the future seem to be the way to go. But we know at the present time, we know too little about their own agendas, appetites, and about the potential harm they can inflict. So this is a painting of Henry Rousseau in our museum in Philadelphia, which gives you a very sort of idealistic picture of a ride on a tiger. Now, the horse is horse. It's not a tiger, but with a horse, you really can do right, which means that red blood cells, which have no other agenda but deliver things from one side in the bloodstream to another. It's a good candidate for a ride. I have 1 more minute. If you don't ask me questions, I will be ruined. Please, please ask your questions. That's it.
Thank you very much for
Please.
this. Yeah. Giuseppe.
Thank you very much. I have actually 2 questions. One is more related with chemistry. You show different, let's say, strategies to get to the target, multivalency or conjugating an antibody to whatever is your target. The question is whether changing something in the conjugation chemistry, like having a linker or not, or changing the position of amino acid, you seem to have some effect in one of your experimental models. That's my first question. And
the answer is absolutely. Absolutely yes. And it does it in a very unpredictable way.
Thank you. And then I would like to, have a little bit your view on what is about the use of immune cells for this delivery. Immune cells. So instead of blood cells, using immune cells, and I'm referring to now, CAR T cells or B-specific antibodies, B cells against autoimmunity. I would like to know whether you ever experienced any kind of therapeutic, let's say, model or like this, or what's your view in general? Thank you.
I don't work with, I do work with leukocytes. We found that in certain situations we can inject ICAM-targeted nanoparticles into animals with induced acute lung inflammation, acute brain inflammation. And these ICAM particles first and very quickly accumulate in the lung, but then a few hours later, they appear in the blood, in the brain. And they appear to deliver there by leukocytes, because leukocytes form very large pool of marginated white blood cells in the lung normally. Apparently there is a relay between endothelial cells and leukocytes, and they can trade goods. And I have no idea how to govern this, because I don't know what the process is. And the short answer to your question is absolutely, but very carefully, because even with red blood cell, I'm presenting things in a very rosy fashion, but even with red blood cells, there is enough problem to be aware of. And the red blood cells do have no other known function but transporting things. And leukocytes have many other functions and not transporting things. Yes.
Tania, aspetta un attimo, ti apro il just technical announcement. You can ask also in Italian eventually, no problem, okay?
Yeah.
If you have questions, not Tanya, you
Okay.
not for you. I mean, in general.
Yeah, and I will answer in Russian.
Yes, and that will be translated.
I don't know if, yeah, okay. First of all, thank you for your really impressive presentation, but I have a lot of curiosities. I'll start with, first of all, which is your best method to anchor single chains or antibodies to RBC cells, first of all, and I have others.
The best method so far is a single chain fusion or a single chain coated liposome or a nanocarrier, a single chain directed to RH17 rhesus antigen. Because when we deliver things to GPA, glycophorin or other very populated red blood cell membrane protein, we inevitably change the red blood cell features. Even if we deliver like 20,000 molecules per RBC, it's enough for these antigens to become more sticky, less biocompatible.
This is another point that I wanted to ask you, especially because you said, we use, for example, single chains for destroy clots, but it was, how can this be possible? You are using single chains, but they actually have this great problem, which is aggregation. When they lose a C domain, they tend to aggregate a lot.
Well, first of all, antibodies, no. They just, we worked with antibodies. And depending on the load, there are some, like 1,000 molecules per RBC, you can load almost anything with any affinity. The body don't detect this. But if you go by a therapeutically reasonable dose, like 40,000 molecules per RBC, or 20,000 molecules, antibodies are no, because they will aggregate. Single chains are different things, especially if you do this with hitchhiking or dual targeting hitchhiking paradigm, because in this case, your affinity to red blood cells is very weak. What's up? What's up?
It's not you.
I remember there was an earthquake in Moscow when I was a student, and we were drunken, and we even didn't feel this earthquake. Only our parents, they say, the chandeliers are moving, but we were thinking that we were just drunk. So single chains, especially if you adjust avidity of your particle in a way that it's reversible binding to red blood cell, then you're fine. Please. Yes.
Yeah, thank you for your very nice presentation, and I will connect my question with Tanya and Giuseppe also about the conjugation technique. Which one do you think, thinking in a translational way, is the easiest to be scaled up to translate a possible technology, drug delivery technology, to the clinic in the future, short or long?
Well, I don't have an authorized answer because I don't do clinical studies. But theoretically, I would say that fusion, single chain, is the best. Not only because it's the most physiologically acceptable. I mean, any conjugation of the membrane will change the membrane. In any conjugation of the glycoproteins like high presence GPA or glycophorin A or band 3, you will conjugate, you will clusterize them. So binding must be monovalent. You cannot do monovalent chemical binding. Because even if you do 1:1 ratio, some will get to 0 and some will get to 2. Fusions and other single chain based delivery systems, they are monovalent. They are risk controlled, affinity, and they are producible, they are scalable. I mean, if I would be an investor, I would invest in the single chains. And also, it doesn't require work with ex vivo isolated red blood cells and reinfusion. You just make the, concoct the recipe. You have it on a shelf, a liposome or LNP coated with single chains. Inject it in a bloodstream. That's it.
How can you, let's say, govern the interaction among the nanocarrier and the red blood cells used as a carrier for the nanocarrier in the term of reversibility of the interaction among it and the endothelial cells, for example?
Well, the game is on the ratio between your primary and secondary ligand. Your ligand for binding to red blood cell is always at very low numbers. I'm even suspecting that 2, 3 antibodies would be enough in single chain. We usually have 5. But for the PKM and IKM, we have 200. Now, I have to be honest with you. I don't think that any company will invest into this super sophisticated, complicated technology unless we will develop delivery for the targets and in the conditions which nobody else can do. Which might be. Please. It never stays the same. It's the same answer as I've done for the whether with chemistry of conjugation effect. The delivery of affinity-based delivery of anything, any target in the body, it's not just a 2D graph of binding versus density or binding versus size or binding versus anything.
Can you ask to use the microphone? There is people connected, otherwise.
Well, they will not be able to see my hands. So it's a matrix. It's a huge matrix in which by changing one parameter, you inevitably change all other parameters. You might have, I mean, the story with PKM domains. You change the specificity of binding site on the same protein, and you can get totally different intracellular delivery. You change the size, everything goes off. You change the valency, everything goes off. It's a huge, well, it's not always black and white, of course. But in many cases, you have to characterize every feature of your delivery system and be very specific. The answer is you change the one parameter, you change everything. But it also works for you, for delivery. Because you might deal with some disease or cell type which nothing gets there, nothing gets there, nothing gets there. And then you change a little bit and it all gets there. And there are some examples of this.
Sono altre domande e curiosità anche.
Ask me in Russian.
I have other curiosities. I'm not an expert in drug delivery system, but I'm quite curious about the nanoparticles you used when your target were in lungs. I mean, I know that actually in lungs we have also the majority of population of macrophages. So I was wondering, which kind of nanoparticles did you use? Because actually I thought, okay, a lot of macrophages, maybe those nanoparticles are cleared very fast. It depends on the, like, I don't know, the charge, the dimensions. Yeah, I know nanoparticles, but which kind of about them?
Well, first of all, it's absolutely right. When you deliver something via the vasculature, you deal with several competing processes. First is, of course, clearance of the bloodstream from your stuff. And if the clearance of the bloodstream is too fast, and usually it's very fast, then you have no chance to get into the tissues which you want to deliver. Now, the lungs in general, specifically, in endothelium in general, is a very specific target. It's very accessible. If you do, I mean, when we do targeting with LNPs with luciferase, we see bioradioactivity, maximal uptake, like 3 minutes post-injection. It's already in the lungs. Now, there is no luciferase signal yet, because there is no RNA produced protein yet. In 3 hours, we start seeing luciferase signal, but the iodine or radioactivity goes down because cells are already dismantling these things and chew and spit out the materials. Cells don't like these polymers. But what your question was?
Which kind of nanoparticles? And did you have like particular attention when you chose the nanoparticles characteristics?
Selection of a nanoparticle depends on the cargo and on the goal. But generally, liposomes are very good nanocarriers for many cargos, including some small molecule drugs, like dexamethasone. LNPs are good nanocarriers for the RNA and siRNA and maybe CRISPR systems. Now, liposomes are relatively uniform. LNPs, there are hundreds, thousands of formulations of LNPs. And you change one parameter, and everything changes. So, nanogels are very good carriers, but they are not good for many cargos. So, it all depends. And of course, you want to have a sufficient circulation for things which you propose to go beyond BBB or in the tumors. And these things need to have a cycle of circulation in order to get to those.
Can I also ask a question?
Oh.
No, I have a question about internalization. I mean, by experience, we have seen that some antibodies on some cells with the same target, but on some cells are internalized, in other cells are not internalized. For example, in EML and versus normal lymphocyte, in one case it's internalized, in the other not. So the question is, as a rule, also what's happened inside the cells in the internalization process?
Well, Mauro, you are too good a friend to sort of try to give you some not truthful answer. The truthful answer is that most of the internalization things which I've showed, and I think most of the internalization thing other people show, is being done in cell cultures. And I don't trust cell cultures. Cell cultures are meaningless in the studies of drug delivery because these cells don't represent their counterparts in vivo. They never sit with exposure for 20 hours in these astronomical concentrations of the particles. Cell cultures are not good. Now, why do I show this? Because there is no way to test subcellular addressing in vivo. There is no technique which gives you relatively in real time, in situ, resolution, spatio-temporal resolution, which would say, these are endosomes. No, unfortunately not. Maybe, I hope maybe in last year and future year or maybe decade, a new tool will evolve. A tool which will be giving you an information of which cell type and maybe even subcellular compartment you deliver. But now, the best system for study internalization, I think, is perfusion of isolated organs. But it's not really the same organ as in our body. We have to understand this. And second, even for perfused organs, we don't have this in situ real-time imaging. We have to have tissue slicing, staining, homogenization and FACS, etc. And all that is semi-informative. I mean, you can publish lots of papers. Sure, be my guest. And when a speaker gives you a point, usually he or she over-exaggerates. There is some information, of course, from these cell culture studies. There is. I mean, they provide you a range of possibilities. The cell culture studies don't tell you what happens in vivo, but they tell you that this might happen in vivo. For example, as Mauricio noticed, the same engagement of the same glycoprotein on the endothelial cell by different domains might lead to different uptake or to different intracellular trafficking. This is sort of an important information because it was without a precedent. Now, it doesn't mean that this particular antigen will behave the same in vivo. I gave you the most honest, self-opening answer.
Thank you. I want to know if I have two quick questions. If I want to know if is there already a strategy to target, in addition to proteins, also the specific composition of double layer of membrane of phospholipids of endothelial cells. And another, okay.
Let's get one by one. I have difficulty skipping even one question. So the answer is yes and no. Yes is that annexin-5 binds to the cells, exposing phosphatidylserine. That's exactly what you mean. Now, would it be of any use for drug delivery? I have no idea, because I don't know whether specificity, yield, efficacy will be sufficient. So, generally, I mean, it's interesting. How did you know? I just started a new project in my lab like 3 months ago where we started comparing conjugating things to glycoprotein on the red blood cells versus insertion into the plasma lemma. And it's interesting, those two approaches to paint red blood cells are very different.
Thank you. Another question is if we can, is similar to the first one, is if there is a strategy to, in addition to target the endothelial cell, also is there a strategy to block the nanoparticle or the carrier inside the cell, endothelial cell, by exploiting the different environment of two sides of the endothelial cell, so the lumen and the I don't know, I don't remember exactly, the muscle cell, muscle layer.
This is a wonderful question. I don't know how to influence endothelial cells from the abdominal surface. Endothelial cell, the general structure of vascular wall, vessel wall is endothelial cell is a monolayer, which is laying on the basal membrane made by endothelial cells themselves, which is collagen 5 and 4. And then, depending on the vessel type, either nothing in the capillaries or a muscular layer of the smooth muscle cells in the arteries, or fluffy interstitium with fibroblasts and some vasa vasorum as in veins. I have no idea how to get there. Now, lumen of endothelial cells is very different in different vessels, and even in the same organ, even in the same vessel, two adjacent endothelial cells might be very different. For example, an endothelial cell sitting in the bifurcation will be different from endothelial cells sitting in the tubes.
Thank you very much.
But I just want to tell you one more thing. Vasa vasorum. Vasa vasorum means microvasculature in the adventitia of the vessels. I'm almost sure, I can't prove it, but I'm almost sure that the most of negotiations between blood and endothelial cells in the large vessels occurs not from the lumen of the large vessel, but from the vasa vasorum capillaries in the interstitial layer of the same vessel. Because in the large vessel, the vascular shear stress and sturdiness of the endothelium are such that everything is working like that to avoid edema. In the vasa vasorum, it's almost like capillaries themselves. That's why I think that, for example, leukocytes and LDLs get to the atherosclerotic plaque not from the lumen of the aorta or the coronary arteries, from the vasa vasorum. I'm almost tired, but if you have more questions, you'll give me more energy.
Ma possiamo approfittare ancora, se c'è qualche domanda prima.
Well, I told today to Sarah that all researchers get grumpy, overcritical, discouraging to anything but their own stuff. Of course, our stuff is great. Don't listen anything which you could feel as a discouragement from my side. You know better what to do and how to do. I'm sure that you will all achieve a lot, and you will decode the things which we even don't think about today. Remember, in 90s, who was talking about extracellular vesicles or exosomes? Nobody. Have a good time.
Yeah, thank you very much for your time, your presentation, your patience with us. I think it's been a very, very productive and useful afternoon. I very much enjoyed, and the presentation, in my opinion, now merit additional time from our part to think about it. I mean, you provide a huge number of information and possibilities, and I think that will be very useful for what we are doing and the job each of us has in the daily life and in the projects we are managing. So really, thank you so much. Thank you very much. Vlad is with us all the day, I mean, up to... I'm
here.
Yeah, so if anybody is interested in asking questions or having a little bit more time with Vlad, I'm sure he will be available for personal or common.
We can put the chairs in the first row.
Yeah, absolutely.
In a circle, and if you are interested, it's not only about science. It's not only about the data. If you have questions of sort of career building or perspective on your future profession, no problem.
Yeah, it will be also interesting to understand. I mean, we have some capabilities, some background, and some piece of equipment that we can use for selected application. But a number of questions of today were also related to activities that are belonging to other groups. I mean, that we don't have in-house in Urbino, in our network. And I think that could also be an issue. I mean, in case someone is interested in understanding which one in the world is the most appropriate group or lab for a specific piece of information because of the expertise behind or in the area, well, take advantage. Vlad certainly know much better than me what's moving in the field, campus, and other areas around.
I don't think that I know much better than you. We just, our knowledge is partially overlapping.
Okay, so formally the presentation is closed, the discussion is closed, but Vlad is there. So if you want to stay, no problem. He is available, he is kindly available. So don't exaggerate, but you can take advantage from it. Thank you, Vlad.